Welcome to this explainer. Have you ever stopped to wonder how scientists actually manage to study the microscopic culprits that are just surrounding us every single day? Well, they do it through some absolutely incredible microscopic detective work, and that is exactly what we're going to be diving into today. We are looking at the fascinating art and really the science of microbiology laboratory staining. But first, we really have to start with the core problem facing literally every microbiologist out there. How do you study something you can't even see? I mean, bacteria are tiny, obviously. But worse than that, they're essentially semi-transparent. The refractive index is incredibly similar to the water they actually live in. So if you just try to look at them under a standard microscope, they practically blend right into their environment. They are completely invisible to the naked eye. Okay, let's dive into this. Here is our scientific roadmap for today. We'll kick things off with making the invisible visible, then move into the chemistry of color and preparing the bacterial canvas. After that, we'll look at how stains actually stick, explore the Gram stain mystery, and wrap up with some advanced staining techniques. Starting with section 1, making the invisible visible. Let's talk about the actual purpose of staining. To unmask our invisible microscopic suspects, we have to get a little creative and manipulate light and color. Because staining isn't just about making things look pretty on a slide. It provides the absolute necessary clarity required to view these specimens, especially under high-powered oil immersion lenses. Basically, it serves three main purposes. First up is contrast enhancement. This produces a really stark color contrast, making those semi-transparent bacteria suddenly pop right out against the background. Second is morphological study. This is crucial because it allows us to accurately study their exact size, their shape, and how they arrange themselves. And third, structural identification. By highlighting specific external structures like capsules and flagella, or internal ones like nuclear bodies, staining is what actually allows us to properly classify these organisms. Moving on to section 2, the chemistry of color. Let's look at dyes, stains, and the molecules behind them. Let's shift our focus to the microscopic chemistry that makes all of this possible. Now, you might hear the words dye and stain tossed around interchangeably, but in the lab, there's a distinct difference. A dye is a general organic compound, often a synthetic derivative of aniline, like coal tar dyes, and it imparts color to almost any substance. But a stain is highly specialized. A biological stain is a specific type of dye used exclusively for coloring biological specimens, like our little semi-transparent microorganisms. Now, what's really interesting about this slide is how these biological stains are actually built on a molecular level. They consist of an organic compound linked to a benzene ring, and they have two vital functional groups. First, you have the chromophore. This is the color-bearing ion. It's the part that provides that beautiful, vivid hue. But here's the catch. The color is useless if it just washes away, right? That is where the oxychrome comes in. This is the ionizing functional group. Think of it like molecular Velcro. It's the absolutely crucial component that allows that beautiful chromophore color to actually stick and bind tightly to the cellular components. So how does this molecular Velcro interact with the bacteria? Well, think of it exactly like a magnet. Bacteria at a neutral pH naturally carry a slightly negative charge on their surface. So if we use a basic or cationic stain, which carries a positive charge, it acts just like a magnet. It snaps right to that negatively charged bacterial surface. Great examples of this are crystal violet or methylene blue. On the flip side, we have acidic or anionic stains, and these carry a negative charge. Because negative repels negative, the bacteria literally push the stain away. So instead of coloring the cell itself, acidic stains, like India ink or nigrosin, color the background, leaving the bacteria standing out as these really cool, clear silhouettes. All right, section 3, preparing the bacterial canvas. Smears and fixation. Look, before a detective can paint a profile of a suspect, they have to meticulously prep their canvas. And in the lab, the very first step is creating a smear. A smear is simply a very thin film of microorganisms spread out over a glass slide. And I really can't emphasize the words thin film enough here. Spreading a tiny, minute sample is incredibly crucial, because if the smear is too thick, you're just going to get massive clumps of bacteria. And those clumps make it entirely impossible to actually discern any individual cellular morphology or see how the cells are arranged. Once you have your sample, the procedure is very active and precise. First, you spread that minute sample onto the slide. Second, you have to let it air dry completely. Then comes step 3, which is a highly critical step, heat fixation. This is where you pass the slide through a Bunsen burner flame several times. Actually, sometimes for really tough microbes like mycobacteria, this even involves heating it at 80 degrees Celsius for 15 straight minutes. Finally, you're ready to stain and observe. This heat fixation step is so vital because it instantly kills the microorganisms, making them safe to handle, and it acts like a powerful glue. It adheres them firmly to the slide so they aren't completely washed off during all the subsequent staining processes we're going to put them through. Which brings us to section 4, how stains actually stick. Let's dig into the physical and chemical theories. Okay, so we know they stick, but let's explore the actual physics and chemistry of why they don't just wash away. There are two primary theories here. Physical theories suggest that staining happens through natural forces, things like capillarity, osmosis, adsorption, or absorption. A great example of this is solubility, where fat stains like Sudan dyes are just more soluble in the cellular fat than they are in alcohol. So the crucial point is that in these physical processes, the dye is just coating the surface. No new chemical compound is formed. On the other hand, chemical theories argue that real chemical affinity is happening. This involves strong electrostatic attraction, like ionic bonding, between the dye itself and the actual components of the cell. But, often the stain just needs a little chemical assistance to get the job done. This is where auxiliary agents enter the picture. We frequently use something called a mordant. A mordant is basically an intermediate substance that actively increases the affinity between the specimen and the dye, helping it bind much stronger. We also rely heavily on decolorizers. These are organic solvents that wash the dye away from some cells but not others. It effectively differentiates the cells based purely on their ability to retain that initial primary stain. Section 5, the gram stain mystery. Let's talk about differential staining. This whole concept of decolorizing brings us to a truly elegant technique. It was developed by Christian Graham way back in 1884. The Graham stain sorts bacteria into two distinct profiles based entirely on their microscopic armor, and it uses this dramatic four-part process. Step 1, you apply the primary stain, crystal violet. Step 2, you add the mordant, Graham's iodine, which forms a complex to lock that color right in. Step 3, you apply the decolorizer, usually 95% ethyl alcohol or acetone. And finally, step 4, you apply a counterstain called safranin. Each step systematically builds upon the last to chemically differentiate our invisible culprits. And this brilliantly illustrates the ultimate result of that four-step process. The Graham stain classifies bacteria purely by the structural composition of their cell walls. Gram-positive bacteria possess this very thick peptidoglycan layer. When the alcohol hits, this thick layer dehydrates and it traps the crystal violet iodine complex inside, making the cells appear a deep violet. Gram-negative bacteria, however, they have a super thin peptidoglycan layer and high outer lipids. The alcohol just dissolves those lipids, the primary stain washes right out, and the now colorless cell takes up the pink safranin counterstain. So you get violet versus pink or red. It is an incredibly powerful diagnostic tool. Finally, Section 6, advanced staining techniques. What happens in extreme cases? Let's see how all this builds when we encounter specialized tools for extreme bacterial cases. Because let's be real, not all bacteria play by the rules. Take acid-fast staining versus vital staining. Certain pathogens, specifically mycobacteria, have cell walls that are absolutely packed with this waxy mycolytic acid. They are nearly impermeable to ordinary stains. So to breach this armor, we actually have to use the physical force of heat and steaming to drive the primary stain directly into that waxy wall. Then, on the completely opposite end of the spectrum, is vital staining. This technique uses gentle wet mounts to study living, viable biological specimens without killing them. So we completely avoid that harsh heat fixation process we talked about earlier. When we perform that extreme acid-fast stain, the results are really striking. The acid-fast bacteria successfully retain that primary stain, carbylfuchsin, and they appear a bright, vivid red. Meanwhile, all the non-acid-fast cells are decolorized and take on the counterstain, appearing blue or green. And it's really important to note exactly why they earn the formidable title acid-fast. It's because once that waxy armor takes in the red dye, they hold on to it so tightly that they resist decolorization even when they are blasted by an extremely strong, harsh acid-alcohol mixture. They hold fast. Which leaves us with a truly fascinating thought to wrap up on today. If a relatively simple series of chemical stains can reveal the invisible, waxy armor of a deadly pathogen, what other unseen microscopic battles are happening all around us right now, just waiting for the exact right splash of color to expose them? The microscopic world is vast, and staining is essentially our ultimate flashlight in the dark. Thanks so much for exploring it with me in this explainer today.